Good afternoon, good morning everyone. My name is Seth Gammon. I'm the product manager of the in vivo line. Today I'm going to be talking about how to utilize our in vivo MSFX Pro for non-invasive imaging of plant physiology using fluorescence and X-ray, as well as introducing the capabilities of the instrument for conducting experiments using luminescent and radioisotopic capabilities as well. So first we'll just take a quick look at what we're going to talk about today. I'm going to introduce you to the in vivo MSFX Pro and then go into imaging by parts of a plant, and then finally end up with spectral unmixing and how that can be important for fluorescence imaging in particular. The question often comes up, why do we want to conduct imaging experiments in general? First of all, you can utilize whole tissue imaging, such as you get from the MSFX Pro, to get better data out of your microscopy or in vitro follow-up experiments, because you can visualize the molecular events that are occurring at the entire tissue level and then just sample the most relevant areas for your microscopy or histology or Western blotting or deep sequencing applications. If you're able to conduct a longitudinal study with your system, then you not only are getting all of that good data I talked about before, but you're also able to measure a true longitudinal history of the molecular pathway that you're trying to study. You're not looking at cross-sectional studies. That actually empowers you to use fewer samples, fewer plants, because you get better statistical power. Also importantly, if you can conduct a longitudinal study, you can track the migration of events throughout a tissue structure. One can think of imaging sciences being very different, but in reality they're all actually very similar. Whether you're looking at the very small end of imaging, such as microscopy, or the very large end of imaging, looking at astronomy, for the most part, you're always imaging light at some wavelength and determining whether that signal is above background of your system. What's different about all of these, of course, is how you interpret that data. What are the different confounding variables that you have to control for? Today I'm really going to focus in on a system that works for both in vitro assays as well as model organism imaging. One of the things I'd like you to take home from this is that the Carestream MSFX Pro is really a one-source instrument for in vitro imaging. You can use the system to do Western blots, including multiplex Western blots, for molecular imaging in model organisms by using the fluorescence, luminescence, and radioisotopic imaging capabilities, as well as a solution for phenotypic imaging, utilizing the X-ray as well as reflectance capabilities. Reflectance is what I'm referring to. It would be like taking a photograph. Here's a look inside the instrument. You'll notice a couple of things. First, we have it's a benchtop system, and you have a xenon illuminator to the left. We'll talk about that more later. You have a housing, including a management area. If you collaborate with people who want to study animals, there are ports for isoflurane anesthesia that can feed into the system. And below you have a unique closed optical path design, which I'll get into more in a second. So let's take a look at how the system works. Over to the left here, you have a 400-watt xenon illuminator. That then emits light through an emission filter wheel. In the MSFX Pro, you have 29 position filter wheel, 28 of which those have very narrow bandpass filters. So you illuminate your target sample, and the fluorophore is then excited. The fluorophore goes up to an excited state, loses a bit of energy, drops down to its ground state, and then re-emits light at a longer wavelength. That hits a 45-degree mirror and then passes through our patented wide-angle emission filters. So in this setup, there's a couple of things that are different from anything you'll see anywhere else. One is that, much like an epifluorescent microscope, we image from the bottom, and that's so that you can much more easily quantify across the entire field of view. If you image from the top, you're looking at different distances from the light emission to the detector, and that will affect your ability to quantify that data accurately. One of the other things that's different is this is a true closed optical path. What that means is that dust, dirt, other things that might harm your optics are completely isolated away between the sample and the detector. That improves the lifetime of your instrument. And last but certainly not least, as I mentioned before, we have wide-angle emission filters. This is something that's very different from fluorescence microscopy in that in fluorescence microscopy, all the rays of the light are basically coming in to the filter in parallel, which means standard interference filters are just fine. However, in a system that uses low f-stop lens, such as the f2.5 lens we have in this system, you're collecting rays from much wider angles. And in that case, interference filters actually begin to fail, and you start to get artifacts that allow excitation light to break through. That means that, again, you get one, poor image quality, and two, you significantly would have your ability to evenly quantify across the field of view decrease. With our patented wide-angle emission filters, it doesn't matter what angle those filters come in, you're able to block that light off. So once the light travels through our emission filters, you enter into the digital camera. The camera itself has a continuous zoom lens, so you can image samples from 2 by 2 centimeters, a field of view, all the way up to 20 by 20. That light is then imaged by a cooled 16-bit digital camera with 4 million pixels. That allows you to get high-quality images as well as acquire for long periods of time when low-light imaging is important, such as in luminescent samples. You switch to radioisotopic mode. There's a phosphor screen that's placed in the instrument. The system then drives that underneath the sample. The sample, having been injected with a radioisotope, such as I-125, then emits a high-energy gamma. That high-energy gamma is then collected by the radioisotopic phosphor screen and re-emitted as visible light. It then enters into the closed optical path chamber just as we had before and is imaged just as you would from that point on with fluorescence, except we don't use an emission filter. In luminescent mode, you have a chemical oxidation, typically from an oxidative species like luminol, which can be excited by hypochlorous acid, hydrogen peroxide to some extent, although it requires high concentrations, as well as systems like luciferase, where you're oxidizing luciferin within a structure. That allows you to look at things like promoter activity and protein-protein interactions. In reflectance mode, this is basically just taking a simple photograph of your sample. You can set the xenon excitation source to the 29th position, which is an open filter, and that lets white light all the way from the visible to the near IR through. It hits the sample and reflects back down. It then goes through in an unfiltered image, or you can collect a red, green, blue image if you want by pre-programming the system to automatically collect red, green, and blue reflectance images and then co-register and overlay them either in the multispectral software or in ImageJ, which is available freely from the NIH. Last but certainly not least is the X-ray system. The Carestream MSFX Pro utilizes a really very thin, very uniform radiographic phosphor screen that comes from our history of being part of Kodak. Kodak is able to make films. That's what they do. They're the best in the world at making thin film technology, and we brought that technology with us when we came away from Kodak. And the X-ray image quality is impacted significantly by the quality of that radiographic phosphor screen. So the radiographic phosphor screen slides underneath the system. The microfocus X-ray head turns on, illuminates the sample. The phosphor screen then kicks off visible light. The camera then detects and quantifies that light just as we had in our other systems. As you notice, the sample itself never has to move as you switch between modalities. So we can directly co-register any of these modalities with one another without requiring any image warping. Essentially, this system is 5-in-1. That allows you to minimize bench space, cost, as well as maintenance, and maximize your results by having the versatility to really conduct nearly any kind of molecular imaging experiment that you might want. So let's move on to some examples of imaging plant and plant parts. I've broken this section into 3 parts: seeds and fruit, seedlings, and leaves and roots. Let's start with seeds and fruits. A very common application one might want to do is be able to screen seeds for from expression of a transgene, such as a fluorescent protein. In this case, people wanted to screen for DS red, and then co-register that with a reflectance image of the seed, a white light trans image of the seeds. So you put the seeds in the dish, place that dish on the platen. First, collect your white light illumination image, then collect a fluorescence image exciting at 535 and emitting at 600, and then these can be directly co-registered and overlaid in molecular imaging software that comes standard with the system. Not only can you count these quite easily by visual inspection, you can utilize the MI software to automatically find areas of peak intensity for you and count the number of transgenic seeds. Using a similar strategy, but with X-ray, you can look at whether seeds are empty or full of soft tissue. Our system utilizes a very high resolution soft X-ray system. The resolution one can achieve is better than 25 line pairs per millimeter, quite easily on contact, and with geometric magnification you can go even higher. In this case, seeds were separated into known pools of empty versus full seeds, and then imaged using our X-ray technology. As you can see, without having to open up or otherwise destroy these seeds, you can quite easily discern the empty seeds from the seeds that are filled with tissue. Next question is, if we throw a random assortment of seeds on a platen, are we able to separate out both visually and automatically the empty seeds from the non-empty seeds, or more likely the viable seeds? And the answer is yes, otherwise I wouldn't be showing you the example. So there's 2 things you can do. First, with black and white contrast, black and white gives the viewer the best contrast. However, when you have a color image, it's oftentimes easier for people to quickly see differences in density, especially over a narrow range. So in this case, you can directly overlay on the same image a color lookup table over the X-ray, or a reflectance image if you'd like, to look at the X-ray density versus the total number of seeds. And then you could count these by hand. More interestingly is to use the auto ROI analysis available in MI in order to count these seeds. So first, we simply set a threshold that for X-ray density that allows all of our seeds, whether empty or not, to be counted. Then you have it search the area for the number of regions of interest, and it does a really nice job of counting these seeds. We then want to just count the ones that are more likely to be viable. So in that case, we want to remove seeds that only have a very small area of high X-ray density. In this case, samples that had less than 400 pixels we were going to reject. You can also, rather than measuring this in pixels, you can measure these in millimeters or squared or centimeters squared, whatever your favorite unit is, because the system is fully automated. It knows what field of view you're at, it knows the number of pixels in the camera and the binning state, and therefore it knows the area. You're able to quite rapidly then reject those seeds that are empty. And in this case, you get 78 coming back out that are full, so the likely viable number is around 81%. And all of that can be done very automatically. Each X-ray acquisition is very short, and then the software analysis can be done in around 10 seconds. You can also look inside of various fruits for looking at fruit quality, and I want to show you this as an example, mostly to highlight another nice feature of the X-ray on this system, which is the ability to use different energies of X-rays to contrast different hard and soft tissue structures. Each of the following images that I'll show you were acquired using exactly the same settings, except we switch an aluminum filter that hardens or raises the average energy. So here's the hardest filter, and you can see that the less dense structures absorb the X-rays less easily. So if you have areas where you have holes, you can quite nicely make those out. As we soften the X-rays, the tissue does a better job of absorbing it, and you see differential contrast building up. On the softest X-rays, you have really nice contrast up here in the leaf structure versus the tissue structure now. So as you can see, by having a system with multiple aluminum filters, it gives you the flexibility to really hone in on just the exact X-ray conditions that you want to use to image your sample. Next, we'll move on to seedlings. This was a group that was interested in looking at GFP phenotype in seedlings. And they used a simple experiment. They captured a fluorescence image of the chlorophyll in a red channel. They then took another fluorescence image in the green channel, exciting at 480 and emitting at 535. And then overlaid the green over the red. And you can nicely see areas of high, medium, and no expression. Another example was someone who wanted to do a very similar experiment, was looking for RFP expression in roots of a cress. And not only were they able to see RFP expression, but there was some potential fluorescence coming from the head here. Unfortunately, they didn't have the multispectral capability. It would have been very interesting to try and do a spectral deconvolution of the autofluorescence while trying to image the RFP at the same time. The same group was also interested in GFP, and they got a very similar result, although you notice that there's much less coming from the leaf structure than we saw before. Here's an example of a group that had a very interesting experiment, which was to not only to use dichlorofluorescine as a way to look at reactive oxygen species. So here you can utilize, again, the autofluorescence coming from the leaf to give you the rough structure or the areas of undamaged structure. And then you can utilize the dichlorofluorescine to look at areas of high reactive oxygen species. Another group was interested in looking at the vascular structure of a leaf. For those people who are familiar with in vivo imaging talks on small animals, unlike small animals, autofluorescence in leaves is high throughout the visible and the near-infrared spectrum, particularly in the red and near-infrared due to things like chlorophyll and cyanines. Phycocyanins. In this case, they still thought that they could use a near-infrared probe to look at vasculature, but they wanted to use spectral unmixing to separate out the plant autofluorescence from the fluorophore of interest. In this case, they utilized indocyanine green, which is a very inexpensive, commercially available near-infrared fluorophore that can be solubilized in water. The leaf was then dipped into the solubilized ICG, and then through diffusion, traffics up through the leaf vasculature. The image was then spectrally unmixed, and you're able to quite nicely separate out the leaf from the autofluorescence in green, the leaf vasculature in red or pink from the autofluorescence or fluorescence coming from chlorophyll in the green. And I'll go much deeper into this later in my section on multispectral imaging. Another very interesting application that's come up recently is looking at host pathogen interactions, particularly in soybean roots, using near-infrared fluorescence. It turns out that the root nodules are, for an unknown reason, autofluoresce strongly in the near-infrared, particularly when looking at a 670 emission. You can then, so you can take a soybean plant, you can infect it, and then put its roots on the platen and image them by both fluorescence and reflectance imaging. On the left here, you get a nice image of the root structure in a full color image. In this case, by taking a 450, a 550, and a 600, a blue, green, and red image, and overlaying them. And you can already start to see visually one or two of these nodules quite simply. However, when you go to the near-infrared fluorescence, the areas that are affected become much more obvious. In this case, they didn't want to do an automatic counting, but rather they wanted to use the magic wand feature to highlight these nodules and then collect structural information about the nodules after that. In fact, a group at the University of Missouri has taken and improved upon this technique where they wash these nodules off the root, put them onto the platen, and then take another fluorescence image, use the automated region of interest tools that I showed in the seed application in order to count the nodules and reject any root debris that comes through. There's a lot of different data you can get out of this type of information. You can look at changes in fluorescence intensity. Obviously, we do quantify that. You can calculate signal-to-noise ratios by looking at a control sample, or in this case, a control area of the image. Using our line tools, you can also calculate distances from the nodule to the root. Next, I'll move on to spectral unmixing. With spectral unmixing, we want to have the capability to do one of two things. One is to image multiple fluorophores at the same time, which is also known as multiplexing, even though they have overlapping excitation and emission spectra. The other reason is to remove autofluorescence. This is primarily what people use spectral unmixing for, frankly. First, I'm going to just step through a very simple experiment that looks at unmixing different overlapping fluorophores in vitro, just to show that the software fundamentally works. So here you see you have Excite 761, Excite 691s, Q dots, and large Stokes shift 640s, all of which are giving signal in the same 96 well plate. So if these were mixed together, you wouldn't be able to discern the difference between them from a single image. What our system does is it acquires a set of images with different excitation filters. You can use a couple excitation filters or many, depending on your application. The flexibility is there for you to choose that. The system then builds up a stack of images whereby now, at each pixel in the image, the software can look down and understand the excitation spectrum observed at that pixel for every pixel in the image. By then calibrating the system based on pure spectra at individual pixels, it then separates out automatically each fluorophore into its own channel, minimizing crosstalk between all four samples. So in particular with plant tissue, why would you want to do that? As I mentioned, unlike in animal systems, chlorophyll autofluoresces quite brightly in the red and the near-infrared. And so removing autofluorescence is important not only in the visible, but also in the near IR. You could also utilize it to track multiple molecular probes simultaneously. However, as I mentioned, most people utilize this just for removing autofluorescence. So let's take a look in more detail at the sample I talked about earlier. To look at ICG, they excited this sample at 750 and looked at the emission at 830. And there's a lot of fluorescence coming all over, not just from the vasculature. In this case, what we did is modeled an excitation spectrum based on just an ICG containing pixel, one of the major vasculature points. And this can be fitted nicely to a Gaussian curve. What you see here in yellow are the observed fluorescence intensities, and they fit quite nicely to this Gaussian. This model is then saved in the software so that you never have to do this modeling again for ICG. We then model the excitation spectrum of the chlorophyll. In this case, you see a really nice peak out here at 670, which intuitively is very different from that you might see in the ICG. The software then uses this knowledge of the model of the excitation spectrum to go through on a pixel-by-pixel basis and separate out the autofluorescence from the ICG fluorescence. So visually, you can already see it in some ways how the software gets this to work. As we started at the lower excitation frequencies, the chlorophyll took up most of the intensity. As we march our way up into higher and higher intensities, the ICG dominates. So after unmixing, you can have a nice overlaid image of your chlorophyll content with your molecular probe, or in this case, ICG for looking at vasculature. You can also just look at the molecular probe individually. Now you get a really nice crisp image of the vasculature once you've unmixed the autofluorescence coming from the chlorophyll. So in conclusion, I hope I've piqued your interest in a multimodality system for looking at a wide variety of plant phenotypes and functional genomic studies, whether you're looking at changes in seed viability, molecular pathways that can be studied with fluorescent proteins or luciferases. You can do automated region of interest analysis if you have a very high throughput application that you're really just interested in counting the number of molecular events that occur in a tissue. And particularly when working in plants, removing autofluorescence is critical. And the spectral unmixing in the MSFX Pro allows you to remove or study plant chlorophyll fluorescence, it's up to you, at the same time you're looking at some other molecular event. So I'd like to thank you all for your attention, and